Hello and welcome. In this lecture, we will discuss about leaf structure and its modification. So what is a leaf? First of all, we will discuss about the structure of leaf. We know that leaves are thin, flat organs and it develops laterally at the node of the stem. Node is the region where leaf arises. Leaves are normally green in color and an important part of the shoot system. Shoot system is a part of the plant which is present above the ground and root system is present below the ground. A typical leaf consists of the following parts. First one is leaf base. Leaf base is a part of leaf where the leaf attaches to the stem. So leaf base attaches leaf to the stem. It consists of two stipules and dicots, and in monocots it masks the stem. The leaf base masks the stem in monocots. Next is petiole. Petiole is a long thin stalk that link leaf blade to the stem. So petiole is the stalk of the leaf. When petiole is absent, the leaf is known as sessile leaf, and when the petiole is present, the leaf is known as petiolate or stalked leaf. Next part of leaf is lamina, or also known as leaflet. Lamina is green in color at its flat surface, and the main function of lamina is to prepares food by the process of photosynthesis. Lamina consists of midrib. Midrib is a vein that runs along the middle of the lamina. It is a main vein of the lamina. The midrib divides the surface of lamina into two, and it produces thinner lateral veins, which in turn give rise to further thinner veins known as veinlets. So from midrib, thinner veins are produced, and from these veins, further thinner veins are produced, which are known as veinlets. These veins and veinlets give rigidity to the lamina. Also, these helps in water transport, dissolve mineral salts, and also they carry away the prepared food in the lamina. In the figure, this is the dicot leaf. This green color, this big green structure is known as lamina or leaf blade. This is the leaf apex. These are the margins. This is the main vein. This is the midrib, the middle midrib. So the midrib divides the lamina into two, and from midribs, thinner veins, these are thinner veins, and from these veins further thinner veinlets are produced. These give rigidity to the lamina. This is leaf base, and in dicots we discussed that two stipules are present. This is the leaf or monocot. So you can see this is the leaf blade or lamina, and it in sheath or mask the stem. This is the nodes from this area, this leaf develop. So these are the leaf structure in monocots and dicots. Next is the leaf venation. Leaf venation is the arrangement of veins and veinlets in the leaves. And generally there are two types of venation. First one is reticulate venation. In this type of venation, the veins and veinlets form a network, complex network, and it is commonly occur in dicotyledons. For example, rose. So you can see here, this is the reticulate venation. Smaller veins form a network. This is midrib, and from this smaller veins, so you can see they form a chain or a network throughout the lamina. Reticulate venation is of two types. First one is pinnate reticulate venation. Here the main veins and veinlets arise from the midribs and ramify or branches throughout the lamina. For example, occur in guava. So you can see here, this is the midrib. From midrib, these main veins are arise, and from these main veins and further veins and these small veinlets arise and ramify and form a network throughout the lamina. So this is known as pinnate venation. The next type of reticulate venation is palmate reticulate venation. Here the principal veins arise at one point and at the leaf base, and it proceed either upward or outward. So they maybe proceed outward up the leaf blade or upward through the leaf blade. So you can see here the veins, the principal veins arise from the leaf base. These either go outwards or upwards. So there are further two types of palmate reticulate venation. First one is palmate divergent. Here a number of strong veins arise from leaf base, and these then diverge towards the leaf margin like fingers from the palm. For example, occurs in cucurbita. So you can see here, this is the leaf base, and this is one strong vein, another strong vein, this is midrib, another strong vein, another, and these diverge throughout the leaf margin, you can see here, just like finger present on the palm. So it is known as palmate divergent. Next one is palmate convergent. Here veins runs in a curved manner from leaf base to its apex. For example, occurs in cinnamon. You can see here, this is the example of palmate convergence. So here the veins run in a curved manner. It arises from leaf base in a curved manner. It goes to the apex of the leaf. So you can see here, this is another. So it is, this type of venation is known palmate convergent venation. The next type of venation is parallel venation. In this, the veinlets or veins run parallel to each other, and it is commonly occurs in monocotyledonous plant. You can see here, these are the veins, so these are parallel to each other. So it is typically found in monocots. Parallel venation has two types. First one is pinnate parallel venation. Here the leaves has prominent midrib, and this gives off lateral veins, and they runs parallel to each other towards the margin or leaf apex. For example, occurs in canna. You can see here, this is a prominent, this is the leaf of canna. You can see the midrib is very prominent, and it gives lateral veins. These are lateral veins. So they run parallel to each other, and they go to the margins of the leaf or here to the tip of the leaf. So in both cases, they either run towards the margin or the tip of the leaf, but they are parallel to each other. And next type of parallel venation is palmate venation. Here veins arise from the tip of the petiole, and either it is also of two type divergent palmate. Either the vein diverges towards leaf margin in a more or less parallel manner. For example, occur in Palmyra palm. So you can see here the veins arise from the tip of the petiole, and they run parallel to each other towards the margin, leaf margin. So you can see here these. The next type is convergent palmate. Here the veins converge from the leaf base to its apex in somewhat parallel direction. Occurs in bamboo. So you can see here, this is from leaf base, the vein arise and run in parallel to each other towards the apex of the leaf. So it is known as convergent palmate venation. The next type is the next topic is types of leaf. There are two broad categories of leaves. First one is simple leaf. Simple leaf is when a single lamina is connected to the main stem by a petiole. It is known as simple leaf. This simple leaf may be incist or lobes, so their lobe will may be present to any depth, but not down to the midrib or petiole. So the leaf may be lobed, but these lobes are not occurs into the midribs or in the petiole. For example, in guava leaves, the next type is compound leaf. Here, a leaf made up of two or more leaflets, and the midrib of the leaf is branched into different leaflets connected by a single petiole. The next important point is axillary bud is present in the axil of the simple or compound leaf, but not in the axil of the leaflet of the compound leaf. For example, palm leaves. You can see here, this is simple leaf. So this is the entire lamina. So it's connected to the stem. Here, axillary bud is present. This is midrib. This is the compound leaf. So you can see here, this is the stem. And it is divided, the midrib is divided into leaflets. You can see here, this two leaflets, other leaflets, and also an axillary bud is present in the axil of the compound leaf, but is absent in the axil of the leaflets of the compound leaves. The compound leaves are further divided into pinnately compound leaf. Here, the midrib of the leaf, which is also known as rachis, is divided into numerous leaflets, all connected by a common axis. And these are further differentiated into unipinnate type. Here, when the midrib bears leaflet on each side of the axis, it is known as unipinnate. The leaflets may be even, which is known as peripinnate, or they may be odd in number, which is known as imperipinnate. So you can see here these two types. Here you can see the midrib. This is the midrib. It bears leaflets both on its side. This is one side, this is another. So two leaflets are present. And these are two types. One is peripinnate, because these are even, you can see even number is present, to here, to here. So all are even in number. This the midrib is known as rachis. Here this is unipinnate, but it is different type. It is imperipinnate, because here odd number are present. You can see odd leaflet, only one is present at the top. So this is imperipinnate. The next one is bipinnate. In bipinnate, a secondary axis bearing the leaflet is produced by the central axis. For example, occur in mimosa. So you can see here, this is petiole, this is the main axis, central axis. Here a secondary axis, this one is produced, and this secondary axis bearing leaflets are present. So on both sides, leaflets, small leaflets are present. So it is known as bipinnate. And next one is tripinnate. In tripinnate, a tertiary axis bearing leaflets emerge from the secondary axis. For example, occur in moringa. So you can see here, this is the central axis. This is the primary axis, also the central axis. This is the secondary axis, and this is the tertiary axis. This tertiary axis have leaflets on both its side. So these are known as tripinnate. Here tertiary axis arise from the secondary axis. The last one is decompound. And decompound type, the leaf has more than thrice pinnate. So here the leaf will be pinnate more than thrice. For example, occur in coriander. So you can see here, this is the leaf of coriander. So it is 1, 2, 3, 4. So it is more than thrice pinnate. So this is known as decompound leaf. So these are different types of leaves. The next type is palmately compound leaf. The one we already earlier discussed was pinnately compound. This is palmately compound leaves. Here the leaflets are attached at the tip of the petiole and can be differentiated into unifoliate. So palmately compound leaf are those leaves which are present at the tip of the petiole. So unifoliate having only one leaflet, bifoliate have two leaflet, trifoliate have three leaflet, quadrifoliate have four leaflets, multifoliate have many leaflets. So you can see here this is one leaflet present at the tip of the petiole. This is two, this is, you can see at the tip of the petiole, two leaflets are present, three are present, four, and more than four is known as multifoliate. So these are different palmately compound leaves. The next topic is Pylotaxi. Pylotaxi is the pattern of leaves arrangement on stem, and generally plants show three types of Pylotaxi. First one is alternate. Here a single leaf develops at each node alternatively to each other. For example, occurs in tobacco. So you can see here this is alternate Pylotaxi. This is petiole, leaf stalk. This is a node area where the leaf develops. So this is one leaf, this is alternatively another leaf, this is another, this is another. So one leaf is present alternatively to each other, so it is known as alternate Pylotaxi. Next one is opposite. Here a pair of leaves develop at each node opposite to each other. For example, occurs in guava. So you can see here, this is very easy. This is one leaf, and opposite to this, this is another leaf. So these two leaves are present opposite to each other. So it is known as opposite Pylotaxi. Opposite leaves are of two types. First one is dicots. Here the two opposite leaves are present at right angles to below one or above it. For example, occur in ocimum. So here the two opposite leaves are right angles to the below one or the above one. So you can see here this leaf, you can see these are 90 at right angle to each other. So it is known as dicots opposite. Next one is superposed opposite. Here a pair of leaves is seen to stand directly over the lower pair in the same planes. For example, occur in guava. So here a pair of leaves stand directly over the lower pair of leaves. So you can see here, this is the lower pair of leaves and this is the upper. So you can see here, this is stand directly over this lower pair. This is stand directly over this lower pair. So it is known as superposed opposite. The last one is whorl Pylotaxi. Here more than two leaves develop at nodes to form a whorl or circle. For example, occurs in Nerium oleander. So you can see here, this is the node area where leaf develops. So at node, more than two leaves develop, one, two, three, four, five, and it form a whorl or circle around this node. So this type of Pylotaxi is known as whorl Pylotaxi. So these are all about different types of leaves arrangement present in plants. Next, we will study about modifications of leaves. Leaves are specialized part of plant and it can perform different functions such as support, storage, food, defense. So it is modified into different structure. These modifications are as follow. First one is leaf tendrils. Leaf tendrils exist in those plants which have weak stems. In this, the leaves get modified into a thread-like structure known as tendrils. Tendrils are climbing organs. It is sensitive to contact and it coil around the contact body and provides supports to the plant. Leaf may be partially or wholly modified into tendrils. For example, in wild pea, the whole leaf is modified into tendrils. You can see here an example. This is the wild pea, and you can see this is the leaf which is modified into a tendril. This is the whole leaf is modified into tendrils, and the leaves like structure which you can see in this figure, it is actually the stipules present and in the main leaf, the axil of main leaves, which is modified into tendrils. In Pisum sativum, the upper leaflets gets modified into tendrils. This is the example of Pisum sativum, also known as pea plant. So you can see the upper or terminal leaflets are modified into tendrils. In Gloriosa, the leaf apex is modified. This is Gloriosa leaf, and you can see the apex of leaf is modified into tendrils. In Smilex, the stipules are modified into tendrils. The stipules present in the axial of leaves. This is stipule, this is the main leaf. So you can see the stipules in Smilex are modified into tendrils. So these are different types of tendrils found in different plants. Next type of modification is leaf spines. Here leaves are modified into a needle-like sharp structures known as spines. It acts as a defensive structure, and it also reduces water loss due to transpiration process. For example, in Opuntia, the small leaves present on the modified stem is modified into spines. You can see here, this is the example of Opuntia. So this big green platen structure is known as cladode. It is a modified stem, and then this stem contains small leaves. You can see here, and these leaves are modified into thorn. So you can see spine. Sorry, these are leaf spines. So the function of these leaf spine is defensive, and also it reduces water loss in plant. Next one is agrimon. It develops spines at the margin of the plant. So this is the figure. You can see this is the agromon structure. So this is the plant structure. You can see the margin contains spines. Next, yucca plant develops spines at the apex. So you can see here in the figures, yucca plant is given and the apex is modified into spines. In Barberry, the modified leaves develop into spines, and the auxiliary bud leaves are normal. This is examples of Barberry. So you can see that the normal leaves develop into spines, they are modified into spines, and the leaf-like structure which is present are actually the auxiliary bud leaves, which are normal in this structure. Next type of leaf modification is storage leaves. Such type of modifications occurs in xerophytic plants and plants present in Crassulaceae family. They have thick and succulent leaves that store water in their tissues, because these kind of plants present in the environment which have water deficiency, so they need to store a lot of water. The parenchymatous cells of these plants contains large vacuoles which have hydrophilic colloids, and it can store a lot of water. Such type of modification helps plant to resist desiccation or dryness. You can see here crassula is given, and you can see the leaves are thick and succulent because they store plenty of water. Next type of leaf modifications is scale leaves. It is thin membranous structure without stalks or brownish or colorless leaves. These leaves protect the auxiliary bud present in their axil. The scale leaves in onion are fleshy and thick because it stores food and water. And asparagus also contains scale leaves. You can see here, this is the example of onion. So you can see the scale leaves are membranous. These are two types. The outer are dry one, the inner are fleshy and thick because it store food and water. This is the auxiliary bud, and these buds are protected by these scale leaves. This is the example of asparagus. Asparagus also contains scale leaves. So this is another leaves modification. Next one is leaflet hooks or leaf hook. In this type, the terminal leaflets gets modified into a hook-like structure that helps the plant to climb. It is found in begonia. You can see here, this is the terminal leaflet. You can see here, this terminal leaflet is divided into a hook-like structure and it helps the plant to climb. The next type is phyllote. In this type, the petiole become flattened, taking the shape of the leaf and turn green in color and perform the leaf functions. It is found in Australian acacia. So you can see here, this is the petiole, the leaf, and this petiole turn into a leaf-like structure. You can see green in color and it performs the function of leaves. The next types of modifications in leaves are insectivorous leaves. In this type, the leaves are modified to catch and digest insects to avoid nitrogen deficiency because these plants present on those soils which have nitrogen deficiency. First one is leaf pitcher. In leaf pitcher plants, for example, Nepenthes, here the lamina is modified into a pitcher-like structure. The insects are captured and digested in it because the inner walls of the pitcher have digestive fluids which digest the insects captures. So you can see here, this is the structure of Nepenthes. So this is the pitcher-like structure. Uh, this pitcher-like structure is made from the lamina. The lamina is modified into this pitcher-like structure and the inner wall of this pitcher contains digestive juices. So when the insect trap here, it is digested. Next one is bladderwort. In this type of plant, the leaves segments are modified into bladders-like structure. The inner walls also contains digestive glands and it digests the insect trap inside. It is found in water. The typical example is Artricolaria. So you can see here, these are the structure. These spherical circular structure are known as bladder, and in the inner wall of these bladder contain digestive enzyme. So when an insect is trapped, it is digested here by these enzymes. Next one is Drosera. Here the lamina contains hair with a sticky globules at the tip containing digestive enzymes. The hair covers an insect when set on the stamina and it is digested. So you can see here, this is Drosera. Here the lamina contains these hairs and these hairs contain sticky globules. So when an insect sits on this lamina, so the sticky globules trap the insect and it is digested here because it contains the digestive enzymes. So these are different types of leaves modification found in plant. Next is functions of leaves. We know that leaves perform some important functions which are as follows. First one is photosynthesis. In photosynthesis, the plants convert carbon dioxide and water in the presence of UV light into glucose. So each and every plant make food by the process of photosynthesis. Next one is transpiration. Transpiration is a process in which removal of excess of water from plants into the atmosphere occurs through stomata. Stomata are the structure present in the leaves. Next one is guttation. Here it is also the removal of excess of water from the xylem at leaves edges by the pores known as hydatodes, and this process occurs when the stomata are closed. So it also removes excess of water, and this phenomena can be easily seen in the morning when drops of water are present and leaves edges. Next one is storage. Leaves also store water and nutrients. It is found in xerophytes. Also, defense. Some leaves are modified into spines and it protects them from damage or and eaten by herbivores. For example, found in Apuntia. So these are all about leaf structure, modifications, and functions. Thank you for watching.